So higher level vision. So the idea is it's a kind of a little bit of a grab bag of different higher level processing in the visual system, because we talked a lot about sort of early edges and motion and relatively simple motion. So I'll start off with color. So if you I mentioned before, when I was introducing the aperture problem, that color, the actual absorption or reflection spectrum of an object, that's what you would sort of think of as the intrinsic color of an object. It turns out that that's actually quite non-straightforward to recover, because the light coming down on an object is all different colors. And you have this when you're sort of inside your system. The system works so well, you kind of don't pay that much attention to it. But if you set up particular color illuminations situations, you can really very easily completely change the apparent color of a patch, and you have to sort of be in these setups where the overall background color can be controlled. So like if you go into a dark room and then there's just projectors that are projecting different colored light onto colored patches, and you look at the colored patches, it's amazing how you can completely change the apparent color of a patch. But the simplest kind of setup which you can do at home is if you take a red light, just like any kind of red light, and a white light. So you just take a red and a white light. This is something you can easily set up, and just on a wall, more or less, white wall, doesn't have to be totally white, but just basically project a red and a white light onto a wall. And then if you actually make a shadow, just stick your hand, you'll get, here's a circular hand. So if you make a shadow, you'll get 3 shadows. So one shadow will have, no light in it, and that guy looks black. But one shadow will just have just white in it, and the other one will have just red. And the background will be, red plus white. But what does that actually look like? So if you actually if you just set this up, a red light and a white light, stick your hand in the shadow. So the black looks black. The red looks red. So this looks very, very intensely red. The background looks white. You won't be able to, if you add a red light and a white light, you won't be able to tell. And it fills up the room. If you're in a dark room and it fills up the room, it looks white. And the white looks green. And I'm not talking sort of just greenish. I mean, it looks like intense green. So that's an excellent demo. It turns out to see all different colors, you just need 2 different wavelengths. They can even be like 2 different wavelengths of green. As long as the objects in the scene are reflecting back varying combinations of those just two green or red wavelengths, you can actually see all the different colors from that, just from different combinations of 2 monochromatic greens. But this one's a little sloppier because you got white light, which has all different colors in it, but it's very easy to set up. So it's amazing. You just have basically the background looks white, and you put your hand in there, and there's this intense green color, which is actually just white light. It's just white light. But because there's red plus white in the background, the system has actually figured out that must be white, and so I'm going to recalibrate all the colors relative to that, and so white will sort of go away from red, which is green. So Yeah, so it's an interesting thing because you can clearly see there's obviously no green light coming from the projectors, but you just have this intense green image. And so if you went into V1, so this experiment has actually been done, not using actual monochromatic light, not the red and white, but basically what the result is, if you go into V1 and you put a V1 receptive field in the white light, you get a signal that looks like white. But if you go to a higher level area, like infratemporal cortex or V4, a V4 receptive field put in the white light that appears green will respond similarly to an actual green. So you could then, actually project just a straight green against a white background, a simple white background, to test that cell likes green, but it will actually like this green as well, and that's the green that's that you perceive when you actually look at this display. So already by V4, it's relatively low level area. It's only like V1, V2, V4. You already have something closer to the actual true perceived color that's doing this subtraction of the background from the local percept in order to actually tell what the real color is. I mean, we're kind of messing with the system here by adding the red light, but basically what is the point of this system? The point of the system is to try to figure out what the intrinsic color of the object is and subtract out whatever the spectrum of the illumination, which can change all over different times of day, shadows. Okay, so that was, so if you want to look up, so that was that experiment in V4 was done by Semir Zeki. It hasn't actually been, there hasn't been too much follow-up on this, but it's an interesting, it's an interesting area. So, okay, so that was some true color. So let's talk about just completely change over to stereo and motion. And one thing, I meant to say this last time, but I forgot to say this last time. So one of the important distinctions about stereo is that stereo is basically a horizontal deviation that differs between the two eyes. So there isn't, you can't make much use of things that vary in sort of in the vertical direction of the two eyes. Really what you're looking at is the two eyes are looking at the same scene, and there's slight deviations in the horizontal position of the object with depth. And one of the interesting observations was that if you look at the left, versus right, Gabor filter in cells in V1. So if you look at vertical orientations, what you find is that the left and the right eye might have a different substructure to the receptive fields. So you might have, say, something like, on in this field and off in this field. But if you look in the, and say this guy is the right eye. But if you look at the left eye, you might find a situation like this, where you've got, say on and off and off. So this is the same cell, but when you look at it, when you test it through the other eye, what happens is the the phase of the Gabor, or just basically sort of the overlap. So I mean, if you put those both on the same thing, if we just draw a line through it that way and then put them on the same graph, what you would see is like the right eye, if this is the middle of it, the right eye might look something like that. And then the left eye will basically be offset. It'll look something like that. So it turns out for vertical orientations, that happens. Okay, they can be the same in both eyes, but you basically have variation in the offset of the filter between the eyes. But if you look at other orientations, basically what happens is whatever the filter is in one eye, it's going to be the same in the other eye. So that's a good piece of evidence that these differences in the filter between the two eyes are actually being used for calculating disparity. Because the main, like I opened up with, the main reason for this is because we're interested in these disparities in the horizontal position between the two eyes. And so if you have filters like this, then you'll have a whole range of estimates of that small disparity between the left and the right eyes. But when you get to vertical disparities, you don't care. You don't really need to measure vertical disparities accurately. You need them a little bit in order to get the eyes. You don't want your eyes going like that. So you want your eyes sort of lined up the same way. So they're not useless, but you don't need to sort of divide them up into so many bins. And so that's what's going on here. So basically, if you look at these, sorry, I drew it the wrong way. If we go across the receptive field perpendicular to its orientation and look at the phase, basically what happens is, we'll just have, if one eye looks like that, the other eye will also look like that. So this guy has a phase diff. This guy has no phase diff. So that's good evidence that what we're seeing here is something that is adapted to giving you a nice range of different, being able to measure a nice range of different horizontal offsets in the image coming into the left and the right eye. Okay, so that was just a kind of something I didn't say last time. But now let's go back to kind of higher level vision. And so one of the things that happens if you move, so say here's your eyeball. There's an eyeball, and and say you're moving your eyeball while looking to the side, and so here's another eyeball. So I moved. I moved while looking out at the world. So I'm looking out at the world, and I moved. So what happens to objects in the world? So say you're fixating here. We have to decide where you're fixating. So say this is your fixation point. So I'm fixating that point. And as I move, I'm fixating that point. Because a lot of times, what could be doing that? Well, it could be something like, optokinetic nystagmus could be helping out, but I might actually just be fixating, say there's something there. Say there's a person here, there's a person here. So so I'm fixating on the person. So this is, fixation point. So there's the fixation point. Okay, but that's just one depth. There's other stuff, and so say there's something, in front here. Say there's a coffee cup Here's a coffee cup in front. And say there's a tree out and back behind your fixation point. So what direction do those guys move? So let me label this guy. So this is observer movement. So if the observer moves that way, and the cup is all these other things are stationary. We assume everything is stationary. What's going to happen is the cup will appear to move this way on the retina. So, because I move the retina this way, so that means the cup's going to go the opposite direction on the retina. But what will happen to the tree? The tree will go this way. So, and you can easily vary. This happens all the time. Again, it's one of these things that's happening all the time. So you don't get a feel for it. But just pick out some object. Like I'm looking out at the chair out there, and then sort of move yourself back and forth. And when I go left, I can see all the stuff on the board going right. But if I look at something that's behind, if I attend to something that's behind the chair while looking at the chair, I can see that's actually moving the same direction as me. So basically, this is, again, if you think of it from a Hebbian point of view, this happens like, the newborn wildebeest already has like, tens of thousands of examples of this correlation. So wherever you're fixating, if your body moves, everything toward you from the fixation point moves opposite to your movement, and everything away from the fixation point moves in the same direction as your movement. So your system obviously could use this information to figure out like what the depth of an object is. So in other words, like if I happen to be fixating here, and I move, anything going to the right, I know has to be behind the object. So there's a very clear association between depth and movement. And so if you go into higher cortical areas, so let me just label this. So this is movement on retina. So this guy's movement on retina. Those are kind of a little bit not dark enough. There we go. Okay, so if you go into the, higher up in the visual system, so if you go in, there's an area called MSTL instead of MSTD. So if you go up into area MSTL, that area seems to be involved with things like tracking objects, and like you're going to be tracking this object in this case because your eye is going to be rotating slightly to keep both of them focused on that fixation point, the face. Put the face in there, yeah. So there's the face. You're looking at happy face. So if you go into MSTL, what you can find are cells that start to put together the streams of motion and stereo. And so an example of a cell, so MSTL, so what you can find is, direction selectivity, gated by disparity. So this is like an example of what we were talking about, where you have these streams, and then but there's no point in just keeping them separate in streams, and so you bring them back together and have them interact with each other. And so what you can find is a neuron that will actually change its direction selectivity dependent on disparity, which is exactly what's going on here. Like depending on the so what's going on here is you have retinal motion. If I boil it down, so retinal motion is correlated with depth. So basically, if it's deep in the scene, it's going right. If it's shallow in the scene, negative, we call that negative disparity, it's going left. And so some of the cells, these cells in these higher visual areas pick this up. And so you can find, something with a response like this, where if the object is moving if you move something this way, and this is zero disparity right in the middle, and this is like minus, these are diopters of disparity, so disparity going this way. Zero disparity would be like at a fixation point, this is zero disparity. So at zero disparity, there's the cell is sort of transitioning, but basically this is a cell that likes leftward movement when it's a cell that would be like this guy. So it likes leftward movement when you have negative disparity, which means disparity that's one way away from zero disparity. That occurs when an object is closer to you than the fixation point. And then when an object is further away from you than the fixation point, it likes the other direction. So this is the amount of disparity. So clearly this is the cell that has kind of learned something like this, basically, that what's ever further away from you than the fixation point moves in one direction, and what's ever closer moves in the other direction. So that information is heavily used for trying to figure out like the shape of an object. Like if an object is rotating, whenever an object rotates, if you're fixating some point in the object, some parts of the object are going to be moving in one direction correlated with depth, and the other parts that are on the opposite side of the object are going to be moving the opposite way. So you could sort of think of what's going on here as equivalent to a rotation of an object. So that's another sort of deep similarity. So what we're doing here is we're kind of, we're moving ourselves relative to the world, but that's kind of the same as rotating the world. Now, it's relatively hard to rotate the whole world, but you could do the same. You can certainly rotate a relatively large object. So an observer movement, equals rotating the world. And so the same kind of argument we have here, we can apply to determining the depth of your scene by analyzing, which direction different parts of the scene are moving. And so that will allow you to tell the shape of an object, if an object rotates. It's sort of similar to how we're detecting the depth of objects in the world because we're moving, which is equivalent on the retina to rotating the whole world. Okay, so I don't know if that was a little bit confusing, but you can begin to see the kind of things that you're going to be looking for in these higher areas where you're starting to mix together all these different cues in order to figure out things like the higher order shape of an object. And eventually, the shape of an object, you get information from all these motion cues and depth cues, but then you also have other cues like shading cues, and there's an overlapping cues, and so there's 20 things that will eventually come together in these higher areas to really build up a robust estimate of what things actually look like out there. Okay, so that was stereo in motion. So now let's just completely change gears again and talk about object recognition. And so an area like V1, it's looking through the world, looking at the world through these tiny straws, and it obviously, it's just seeing tiny little ripply oriented ripples. And that's, and maybe sort of measuring some kind of stereo offset in those oriented ripples, but, it's a long haul up to a face or some kind of complex object. And so it was known from a long time ago that infratemporal cortex was involved in object recognition. And so let's draw where infratemporal cortex is. So if we look at cortex here, so infratemporal cortex is basically, so in a macaque monkey, infratemporal cortex goes all the way to the temporal pole. And so all of this stuff, plus a whole bunch of stuff underneath. And there's even more stuff underneath in humans, because a lot of the, as the brain expands, a lot of areas tend to sort of move around the back, partly because we have sort of more non-primary cortex, which is kind of located here. And so the areas tend to sort of move around the back. So compared to a monkey, there's more stuff underneath. A higher proportion of the cortex is kind of underneath here. So infratemporal cortex, originally it kind of referred to stuff completely underneath, but we know from animal studies that a lot of this cortex on the lateral surface, but below the STS. So here's the STS. So below the STS is visual infratemporal cortex. And so in the very early days, like, probably the 30s, they did some experiments where they damaged this. And there was originally something called Kluver, so Heinrich-Kluver Bucy syndrome. And basically what they did was they damaged infratemporal cortex, but unbeknownst to them, or they weren't paying that much attention, they took out the amygdala also. And so what they found was a strange syndrome where the animals they suddenly became completely unfearful of humans, because a wild monkey has good, well-placed fear of humans, those other primates. So they became completely fearless, but then they also had trouble recognizing objects. And so eventually they did a more discrete lesion and figured out actually it's the amygdala was the thing that was causing all these social fear-related differences, and the part that was causing the object recognition problems was infratemporal cortex. So infratemporal cortex, if we make a drawing, sort of a rough drawing of, so here's V2, and we've got another area in front of V2. It's a little confusing exactly what's going on right in the exact center of gaze, because this is the center of gaze of V1. So there's the horizontal meridian of V1. So this is V2 minus and V2 plus, and then here's V1 plus and V1 minus. So there's another area in front, we'll call it VP plus, and then up here, there's another area, this is in a macaque monkey I'm drawing, called V3, that's minus. And there's, additional areas that we talked about past here. But let's just talk about an area that is, in the macaque monkey, is sort of sitting about here. So this area, it's probably got two areas in it, but we'll just say that there's one. And so this is V4 minus and V4 plus. And there's another little area in here. I won't label that guy. But if you go a little further forward, you get into posterior infratemporal cortex. So there's actually another one down here, but we'll just say PIT dorsal. Okay, so that's posterior infratemporal cortex, dorsal part, because there's a ventral part down here. And if you keep going forward, there's yet more infratemporal cortex, central infratemporal cortex, and if you go, even further forward, there's, anterior infratemporal cortex. So, and there's additional areas down here that they weren't looked at in the particular study, and I'm going to show you. But there's a whole series of infratemporal cortices that are all sort of splayed out here. I should have drawn the other hemisphere, but this is a left hemisphere and this is a right hemisphere. So this is LH, RH. Okay, so what's the point of doing this? Well, there's one very useful set of injections that had been done. So if we inject one color into lower field V4 and inject a different color dye into upper field V4, and then we can go and look at where the connections that are coming into that are. And so one of the things we'll find is, V2, there'll be a spot in V2 that projects into there. And there'll be a spot, in V2 coming from the upper field that projects into there, and there'll be, another spot coming from VP, and another one from, we won't get much from, this connection is quite small, from V3 into V4. There's a little one, but it's very small. But now if we look higher up, what we're going to see are back projections. And what we can do is we can look at the topography of those back projections. And so what you'll find is that there's a back projection coming from posterior infratemporal that's going to that spot. And then there's a back projection coming from posterior infratemporal that's going to this spot. But interestingly, if we look at what's going on in this sort of central infratemporal cortex, what we see instead is just kind of like it looks like a bunch of patches that are all kind of mixed up. So these guys are, this little patch will go up here, but then right next to it, there'll be a little patch that's going to here. Okay, I should have drawn that a little bit bigger. But the bottom line is that this guy is still retinotopic. But these areas up here appear to be non-retinotopic. So they may actually have lost a visual map. And why would that be a good thing? Well, that might be a good thing if you're trying to actually kind of recognize an object no matter where it is on the retina. So so the retinotopy seems to break down somewhere like around central or anterior infratemporal cortex, and those would be sort of sitting like, in a macaque monkey, anterior infratemporal would be sitting right here. Sort of AIT and central infratemporal will be here and also underneath. So people have gone there very early on. Since they knew it was involved in object recognition, they went there very early on to try to, all these are obviously retinotopic also. So they went there very early on to try to record and figure out like what is driving these cells. And so they So back in the 60s, initially, they're sort of trying to figure out like what actually causes the cells to fire. And it was back in the days before, good computer graphics. And so what did they have? Well, they had their hands. They had like a laboratory bottle brush, sort of just junk lying around the lab. And so what they found was that, so one of the things they just found by accident was, they're showing little spots around like Hubel and Weasel, and the infratemporal cortex cells could care less. They go, yeah, but little response, almost nothing. And then they just discovered like a little shadow of a hand on the screen gave a huge response. And so very early on, people, and then they would try like the laboratory bottle brush that doesn't give a good response. But some other cells, they really like the laboratory bottle brush. So that was kind of like the start of sort of the grandmother cell idea that you have these, cells that are recognizing all these different complex patterns like faces and hands. And so over the years, people did a lot of experiments like this. My favorite one was this one in Tanaka's lab, Tanaka and Sato's lab, where they went to the toy store and bought about 500 little toys, and then they glued them onto these sticks. So they had like Mr. Potato Head. We can't say Mr. So they had Potato Head on a stick, and then they had like a little corkscrew on a stick, and all these different little cube on a stick. So they just had a giant barrel of 500 objects, physical objects. This was, and so then what they would do is go in and record somewhere in infratemporal cortex, and then just grab one of these things and put it up there. You go through like 10 or 20 or 50 objects, and I think they actually had some real objects also, like a real potato. Monkeys like potatoes, so an actual physical potato on the end of a stick. And so quickly test with a whole bunch of real 3D colored objects what the response was. And then there was an army of graduate students with scissors and paper. And so what they could find is that sometimes, so say you find a cell that likes a shadow of a hand. So what you could do is you say like, well, what is it really like? Well, does it like something like, if I make something like this out of construction paper, and color this all in, is that good enough? Or, is it good enough to just have this? Is that good enough? I don't need the palm of the hand. Because the thing is, the problem that we have is that there's more possible visual patterns than atoms in the universe. There's just like an unbelievable number of possible configurations of retinal images. And so it's really quite hard to figure out. I mean, maybe it's paying no attention to this at all, and it just wants something that, it just wants something like this. Maybe that's good enough, And so the problem is it's really a difficult problem to do this. But what they did was by doing these little paper cutouts, they found that they could often actually get quite a good response just from a very simplified construction paper cutout of that looked sort of like one of these objects. And then furthermore, they found that these, if you looked through the cortex, so if you looked through the depth of the cortex, they found that these sensitivities were kind of columnar. So you'd find like a little column that likes, say, a shape like this. But wherever you recorded in that column, it would like that shape. And if you went over to the adjoining column, you'd find something that would, maybe like the potato. So that was pretty convincing. It wasn't just an accidental thing. There was a whole series of series of neurons at different depths in the cortex that actually liked these simplified objects. And some of the simplified objects might work out something like this. There'd be like a light square here with a little thing sticking off, and then kind of a dark square. Very simple, something like that. Or maybe, a relatively large thing like this with a small dark thing sticking up. So relatively simple, two-dimensional shapes of light and dark and color is apparently what inferotemporal cortex likes. So that's kind of odd, because you'd figure that by inferotemporal cortex, shouldn't we, we're almost near the top of the system, shouldn't we actually have objects, like real objects, like just a hand and nothing but the hand, or just a hand, independent of what angle you're looking at it. But it turns out that when this has been, redone more recently, it seems to be the case that what you have in inferotemporal cortex is primarily view-dependent. So view-dependent responses. So they're not actually responding to an object in the sense of, I'm responding to this marker, whether you've got it like this. Well, marker's not a very good thing. So, I'm responding, you need more of a three-dimensional object. Yeah, so I'm responding to this umbrella, whether it's actually held like this or held like this. So the inferotemporal neurons, actually, if you find one that likes an umbrella like this, it's not going to like it like this. And so this was done sort of very carefully with these series of objects. This was something that Nikos Logothetis did back in the day. And these were objects they got from the MIT AI lab. There were computer graphics. I call them like paper clips. So they look like sort of these arbitrary sort of three-dimensional cylindrical objects. And so they were sort of, computer graphics generated things like this. So that's the light coming down on the top of it. And that guy's very well lit from the top. That guy's very well lit. So they're like little arbitrary, paper clips, but they're made like thousands of them, and the animal saw a lot of these paper clips from all different kinds of angles, and they had to do a task matching paper clips. So like you'd have another rotation of the paper clip, and the animal had to say, is that the same paper clip? So it was kind of a 3D mental rotation task for the animal. And so the animal was very familiar with all these different paper clips. But we had a whole bunch of them. And so then, after the animal had been trained up, seeing all the paper clips, they went into inferotemporal cortex and recorded. And what did they find? Well, they found almost invariably these roughly sort of cosine tuning curves as a function of pose. So here's 0 to 360. So this is the pose. So if you have imagine you put like an axis down the middle of that paper clip like that, and then rotate that paper clip around the axis, and then look at what the cell response is. And so what they invariably found, so IT cell response. They invariably found is that it would respond to this particular paperclip. The animal, of course, knew that paperclip very well. It had seen it hundreds of times, thousands of times, and it had compared it to other paperclips and determined that that wasn't the same paperclip. So it knew that object as an actual object. But when you actually look at an individual cell, it just responded to this particular view of the paperclip. And if you rotated the paperclip off axis, the response would go down until when you got to the the opposite orientation, there was almost no response. So it clearly was not responding to the object as an actual sort of well-defined object entity, even though the animal could easily identify that object. So that's an interesting point, that most of the cells up here don't appear to be really object sensitive. They're still basically view-dependent responses. And probably the most famous single optical recording experiment done on that was this one also done in Tanaka's lab, where they put an optical recording. They recorded from a little patch of infratemporal cortex, and what they found was if you show faces, actually with three-dimensional, it was a mannequin head. I don't know, for whatever reason, they used a bald female mannequin head. And so here it is from the side. Here it's, starting to rotate. So there's from the side, starting to rotate around a little bit, so you can start to see the other eye. Here it's like, frontal, and then, starting to go the other way. Still see that eye a little bit, and then now all the way to the side again. So if you just took that, it was actually a physical plastic head, and rotated it around, what did they find in the optical recording? Amazingly, they found a little spot of activity that was slowly translating across the cortex. So, like this view would go there, and this view would be overlapping in there, and this view would go there. And so this is the, Tanaka lab. I put it in the reading so you can actually look at it. Tanaka lab, optical IT recording. So they didn't find this for other objects. But if you think about this experiment here, so this experiment here is, this is very consistent with that, which is that you've got, imagine you were recording from a cell that happened to be right here. If you're recording from a cell that happened to be right here, as you rotated the head, you would get a nice sort of cosine type tuning curve, and as you started to get to a pose of the object that the cell didn't like, because it actually didn't match the approximate two-dimensional shape of the object, then the response fell off. Now, whether it's, they tried a couple other objects, and they didn't seem to work. They also did another test, which was just translate. So if you find a cell that likes a face, say like a side view of the face, what you can do is you can do another experiment, which is, just translate. And if you do that, nothing happens. So, translate same pose of face across the receptive field. So if you've got the receptive field here like this, what you could do is, of that region, you could just say, let's just, take a face, a frontal face, and just move it across the receptive field. So in that case, no diff, no response diff. And so it's not sensitive to the exact retinal position. It will always respond to the face as long as you have the right pose. But if you rotate it, then it starts to say, I don't like that pose. So that's some of the, up-to-date, down and dirty evidence for what's going on in infratemporal cortex. So it appears to be non-retinotopic, but so that in this case, like you move the face around, you don't get any difference in response as long as it's the right it's aimed in the right pose. But then when you start rotating objects, their two-dimensional view looks different enough that the neuron doesn't like it. And so you still have neurons that are basically just sensitive to two-dimensional views of objects. And so they must be assembled somewhere else. Are they assembled in any one place? That's a good question. Who knows? Maybe not. Maybe all you have to do is just wire up all these views to the motor system, and maybe the motor system can just sort of use whatever views it needs. Maybe the motor system assembles things into complete objects. It's strange. It's not what people were expecting. And so that's why you might not have even heard of this as much. Now I put one last reading, cool reading, where they tried to essentially use some deep learning network to adapt stimuli in real time to drive the cell more strongly. So basically you show a stimulus, record the response of the cell. A network tries to generate a new stimulus and sees if it gives you a bigger or a smaller response. And if it gives you a bigger response, then keep going in that direction. And so they use like a sort of an object recognition, deep learning object recognition network to generate these things. And what do they get? Well, you get these strange things that really drive the cell well. They're just like this weird little connection. Sometimes it looks like it has a few eyes in it or something like that. You get some, it looks like sort of almost like a face, but not really. You'll get some like strange little pattern like that, and that drives the cell, really strongly. So you can take a look at that reading to sort of see what some of the stimuli are that they evolved in real time in order to actually drive the cell. This was Margaret Livingstone did this et al. You can take a look at that in the readings. Okay, I think I'll end there, David.